Hello students, so let's talk about monogenic versus polygenic. We just finished this up in class. So monogenic means mono meaning one, genic meaning gene. Poly meaning many, genic meaning gene. So we've got one gene versus many genes. How did this look in class? So monogenic traits are traits in which it's very obvious that there is a dominant and recessive allele to each trait. So when we crossed our fingers and put our thumbs over each other, some of us cross the right thumb over, some of us cross the left thumb over. From my class, I think the data said there were 17 left and 6 right. This would seem to mean that the left was dominant, and in fact it is a dominant trait. Crossing the right over is a recessive trait. If you had the dominant trait, you could have alleles capital L, which mean you're homozygous dominant, or you could have capital L, small l, which means you're heterozygous dominant. If you have recessive like me, you've got 2 small l's, which would mean that you are homozygous recessive. So thumb crossing is an example. Sickle cell anemia, we talked about that already. You have a mutation for sickle cell, causes you to have crescent-shaped blood cells because of a protein that was changed. Lactose intolerance is a monogenic trait. We talked about that. You either have the ability to continue to making lactase or you do not. And then other diseases are seen as monogenic traits. Polygenic traits now are traits that it isn't obvious that there is a dominant and recessive. There is, but it's not obvious as to which is the dominant and which is the recessive. So this would be an example we did of arm span. My class only had a few lines, but you may have seen, depending on how you graphed it and what the trait and the arm span was over on the x-axis here, you would see this graph that looked like a bell curve or a curve. And that's because there's multiple genes that are affected. So I drew right here that there's different chromosomes. I don't know how many chromosomes affect arm span or height or eye color or skin color, but they're influenced by multiple genes. So for an example, if we're going to say the taller you are, the more dominant you have, then you might be a person that has, if it's on 3 genes, big A, big B, little or big C, big C. If you're a very short person, you might have the total recessive quality. But maybe you're in between. Maybe you have a little bit of this and a little bit of that. So those are all combinations. When we talk about Punnett squares, you can use a Punnett square to find monogenic traits only. So a Punnett square, remember, is when we take our trait for thumb crossing. So I'm going to cross a homozygous, somebody that can cross their thumb to the left over, with a heterozygous. When I go to cross, I'm going to bring this L down, this over, this down, this over, this down, this over, same thing here. And so I actually get 4 people that are considered homozygous, recessive, or LL. This is also meaning that you've got, that would be my genotype. My phenotype would say that I would have 4 people that cross over with their left thumb. That's how you do a Punnett square. This only works simply with a monohybrid cross with monogenic traits. Polygenic traits would have to have more complicated crosses because it is on more than one gene that helps influence.